few months, and I'm always there to support them. And I like to thank the team, Jivendran and Tizia and the chairperson, and for Hugo, he's already been a big president, and he has been supporting and leading the whole team. And I'd also like to acknowledge Dr. Shubhas, who has made this pathway of training all the students, and he did a wonderful job in training them. And all the team members, Praveena, Teasing and Kalai, Sumitra and Yadavan, Gidanjali, Vayang, Kaizen and Naveen. I think there's a big team behind it, and they made it wonderful, and they have followers all over the world from our YouTube channel. And so I think it's today's topic on insight to the non-coding RNA is very relevant, and in the genomic era, in the COVID pandemic. And thank you, Dr. Siti, for supporting us. And I hope today's webinar will be very interactive and will be useful not only for our students, to the whole world. Thank you.
Thank you very much, Professor M. Ravichandran, for your inspiring opening remarks. Next, I would like to call upon Organizing Committee Chairman, Ms. Fong Ziyan, to say a few words and introduce the moderator of today's webinar.
Okay, thank you, Ms. Montaj. So, greetings of the day to all the participants of the webinar. My name is Fong Ziyan, and I am proud to tell you that I am a third-year student of the BSc Honors in Biotechnology degree program of AIMS University in Malaysia. It was an honor to serve as the chairperson of the organizing committee to organize this webinar. Today's speaker, Dr. Siti Aminah Ahmed, who is a part in the RNA bioresearch group at Infectomic Cluster at One Medical and Dental Institute, Universiti Sains, Malaysia. The Dean of our faculty, Senior Professor M. Ravichandran, who is the Dean of our faculty, that is the Faculty of Applied Sciences, the ABSA advisor, Mr. Jivendran, and all the participants of this webinar. I welcome you all on behalf of the organizing committee. So this is the 8th webinar of our ABSA Webinar Series. It is organized by the AIMS Biotechnology Students Association, also known as ABSA, and we are proud to host it. So thank you, Dr. Siti Aminah Ahmed, for accepting our invitation and becoming a part of our webinar series. So, and then thanks to the Dean of our faculty, Senior Professor M. Ravichandran, for accepting the invitation of this webinar. And I want to extend my thanks to Mr. Hugo, the President of ABSA, and Ms. Vivian, the Vice President of ABSA, for facilitating organizing this webinar. As the chairperson, I would like to express my sincere appreciation to every member of the organizing committee for working hard to organize this webinar. Last but not least, I want to thank Mr. Jivendran. Webinar organizing committee members are very lucky to have him as the advisor for ABSA. His support to ABSA is genuinely appreciable. Once again, I welcome you all and thank you for joining us. So, Ms. Montaj, over to you.
Thank you, Ms. Fong Ziyan. Now I request the moderator of the webinar, Ms. Praveena Paramashivan, to introduce the speaker and start the section.
Thank you, MC. Hello, everyone. I'm Praveena, Year 1 Bioinformatics student from AIMS University. Thank you for giving opportunity to serve as moderator for this webinar. I want to thank all the participants for joining us. We are lucky to have speaker from Universiti Sains Malaysia, Dr. Siti Aminah. Dr. Siti Aminah obtained her doctorate from Universiti Sains Malaysia. She joined USM since 2006 as a research officer, currently working at the Infectomic Cluster, Advanced Medical and Dental Institute, USM. She is a member of the RNA bioresearch group that focuses in the area of RNA biology, including RNomic identification and functional characterization of non-coding RNA and infectious agent, development of molecular diagnostic method based on new ncRNA gene markers and RNA technology. Throughout her research life, she has received multiple university and governmental grants. So far, she has published about 12 publications in peer-reviewed journals with each index of 5. Now, I would like to invite Dr. Siti Aminah Ahmed to take control of the virtual floor. Dr. Siti, the floor is yours.
Okay, thank you, Ms. Praveena, for the kind introduction. And first of all, hi everyone. Thank you for having me, inviting me to join this as a speaker. A very good morning to Prof. Ravi, the Dean of the faculty, and congratulations to the ABSA for the success of having this webinar. So I now I will share my slides. Okay, can you see?
Yeah. Yes.
Okay. Okay. My talk is an insights to the non-coding RNA world. So as mentioned just now, I am a research officer from the Advanced Medical and Dental Institute, Universiti Sains, Malaysia. Okay. These are the outlines of my talk. So it's going to be very brief, not too much on the theory part, and most of it will be the work of my supervisor, Prof. Tang Tian Hock. So I'll touch a little bit about the theory in the beginning, and then goes on with the work that we did in AMD. Okay. So, okay, what is RNA? RNA stands for ribonucleic acid. So it is, unlike DNA, RNA is a single strand polynucleotide chain. So the nucleotide present in RNA have OH on carbon number 2 and also pentose sugar, and have uracil instead of thymine on the nitrogenous base. So central dogma states that the pattern of information that occurs most frequently in cells is from existing DNA to new DNA, which is also called DNA replication, from DNA to new RNA, which is also called transcription, and from RNA to make new proteins, which is also known as translation. However, the central dogma doesn't always apply. Okay. So for a long time, little was known of the dark matter or junk in the genome. So you can see from this bar chart that the percentage of sequences coding for protein increases as the complexity of the humans and eukaryotes increase. So over 90% of the human genome is actively transcribed, but with only about 2% of the genome being protein coding. So a lot of this dark matter or junk was not known before. So people say, oh, okay, this is junk, and they throw it away in the very beginning. So after some time, people do research and more research, and they found out that actually this junk is consists of known protein coding sequences, and it increases with the complexity in eukaryotes. Okay. So types of RNA. RNA can be divided into mRNA and also non-coding RNA. mRNA, or known as messenger RNA. Okay. And non-coding RNA can be divided into housekeeping non-coding RNA and also regulatory RNA. So housekeeping RNA consists of transfer RNA and also ribosomal RNA. On the other hand, regulatory non-coding RNA can further be divided into long non-coding RNA and small non-coding RNA. Long non-coding RNA have nucleotides have more than 200 nucleotides, whereas small non-coding RNA have less than 200 nucleotides. Okay, so small non-coding RNA can further divided into 5 categories here. So the first one is microRNA, which is 19 to 22 nucleotides. That's why it's called microRNA. It's very, very short. Small nucleolar RNA, small interfering RNA, small nuclear RNA, and PV interfering RNA. Okay, so this is the organization of non-coding RNA in eukaryotes. Non-coding RNA is transcribed by RNA polymerase 2 or RNA polymerase 3. So transcript is either monocistronic, which is a single one, or polycistronic, more than one, which is sitting one after the other, or also called intronic, which is in the spaces of intron. So the intronic, and also there's a repeated introns here. So different in the organization of non-coding RNA in prokaryote, we can find small non-coding RNA in this region. So either in intergenic region, so it is called small non-coding, small intergenic RNA, or it's overlapping in the sense direction with the open reading frame, or also it can be found, it can also be found complementary to the open reading frame, which is also called the antisense. It's going the other side from the mRNA, so that's why it is called antisense. So there are various RNA functions here. You can see it is involved in translation. So the sRNA that is involved in translation is ribosomal RNA and tRNA. Transcription, export to protein, RNA processing, RNA editing, splicing RNA, replication, gene silencing, RNA editing, and also it involves in diseases. So meaning that the RNA that is known as junk before has actually functions in the genes. So research was done, and people know now the mechanism of sRNA. One of it is translation regulation. So on your left-hand side, you can see that sRNA is involved in translation activation. So here, small RNA, when it at first 5' UTR covered the ribosomal binding site, so the ribosome cannot go and sit on the ribosomal binding site. Therefore, there's no expression or no translation. But when small RNA comes and bind to the 5' UTR, it opens up. And now the ribosomal binding site is available for the ribosome to sit, and the translation will happen. So in this way, the small RNA helps in translation activation. So different from translation activation, small RNA can also be involved in translation inhibition. So the first part is where the ribosomal binding site is available, but when small RNA come and bind to the ribosomal binding site, now the RBS, the site is no more available for ribosome to sit. Therefore, translation will be inhibited. Okay. So this is the second mechanism of small RNA regulation in the genes, which is involved in transcription regulation. The small RNA, which is in red, as a stem loop structure in red here, which is the small RNA, binds to the mRNA, which is in black, and this resulting in the formation of early transcription stop. So as a small non-coding RNA also was found involved in diseases. So I put here some publications regarding small RNA involvement in diseases. Okay, so you can see non-coding RNA molecules as potential biomarkers in breast cancer, and also the downregulation of BC200 in ovarian cancer. Okay, so many research, more and more research has been done, and more information on the function of small RNA. Now, research questions that usually people will ask. Okay, so how many sRNA are there, and why are they important? And what are their functions in the gene, in the cells, and what is their role in diseases? So to get the answers, complete information on sRNA in the genome need to be gathered. A localization and the organization of small RNA in the genome, we need to know all this, and also the characteristics. So how do we go about to get all this information? So the current methods here, I listed out. One is either experimental approach or the computational approaches. So by experimental approaches can be divided. We can do in two ways nowadays. Last time when the work started, they did conventional sequencing. Okay, they have to build cDNA library manually, and then after that send for sequencing, select the clones, and send for sequencing. But with the technology now, we have RNA-seq, high throughput sequencing. So like this, we can get more information from the by using experimental approaches. So from the data that we get from either conventional sequencing or high throughput sequencing, the data analysis can be done, and from there, small RNA candidates, potential candidates can be annotated. Okay, on your right hand side, computational approaches is by taking, because there are so many now, so many researchers have put all their data on the net. So you can also download the sequences that people have put on the internet and can do comparative genomics. So this is the computational approaches. So the same thing, it also goes to the annotation using so many free softwares that can identify the small RNA. So meaning that the potential candidates can be annotated. So after that, after we get all the potential candidates, we have to do validation either by northern blot, real-time PCR, or microarray. And after that, we test the functional do the functional characterization, or we'll predict what's the function in the genes. Okay, so there are many assays that can be done to characterize, to know what's the function of the small RNA. Okay. So like I said in the early just now, most of this work was done by my supervisor when he did his PhD in Germany. So like this, these are mostly his team publication, identification of small RNA in model organisms, like Archaeologus falgidus, and mostly archaea. His work is mostly in archaea and also E. coli. So when he come back to Malaysia, he started to work in AMDI, and he started to have this RNA, but he lead the RNA biology group. So his main findings was, main finding from this research is the identification of small RNA that is transcribed from the repeated sequences, yeah, which is now known as trans-activating CRISPR RNA, and this contributed to the CRISPR-Cas9 genome editing tool. Okay, I put here his paper in PNAS. Okay, and when he started the RNA biology group, so we tried to identify small RNA in pathogens like Salmonella typhi, Plasmodium falciparum, Vibrio cholera, and the other two, Mycobacterium tuberculosis and Leptospira interrogans. So Salmonella, Plasmodium, and Vibrio the work was done by my colleagues, and the other two, Mycobacterium tuberculosis, was done by my friend, her PhD, and we have identified 1,254 small RNAs in Mycobacterium tuberculosis, and in Leptospira interrogans, which is my PhD, we identified 2,000 over small RNA. Okay, so in addition to the identification of small RNA in Mycobacterium tuberculosis and also Leptospira, we also map the transcriptional start sites in both MTB and also Leptospira interrogans. Okay, by mapping the transcription start sites, we can further divided the small RNA by its location, because now by mapping the transcription start sites, now we can map the 5' UTR, okay, the 5' UTR and the 3' UTR of the gene. So the small RNA here is divided by looking at the location of the small RNA, where it is located in the gene. Okay, so there are 5' UTR derived and 3' UTR derived small RNA. There are also antisense to 5' UTR and antisense to 3' UTR. And there's also antisense to coding sequence. There's intergenic small RNA, where it sits in the intergenic region, like this. And there's intercistronic small RNA and small RNA with overlapping features. So here we show that the small RNA in different growth phases, the expression of small RNA, meaning that this is the validation after we have the potential candidates of small RNA. We do some validation and by running northern blot. So we have different growth phases. You can see that, you know, the expression of small RNA, sometimes it's different in different growth phases, meaning that when the bacteria grow in different growth phases, it needs different kind of small RNA. So then we know that, you know, if the small RNA is expressed in the this growth phase, meaning that it is important to that bacteria at that point of growth phase. So in this table, I showed the small RNA identification in pathogens like Plasmodium, Salmonella and Vibrio, and Mycobacterium tuberculosis and Leptospira interrogans. You can see there's quite a number of new small RNA that we identified. And of course, it shows that, you know, different small RNA is important in different growth phases. Apart from identification of the small RNA in different growth phases, we also did some research and apply the small RNA in diagnostic PCR. So because we found that some new small RNA exist in certain certain types of Salmonella typhi or certain species of Salmonella, and also Mycobacterium and Vibrio. So we did select some small RNA. For example, here we showed that STYR3 is one of the small RNA in Salmonella that can be used to differentiate between the species of Salmonella. The same goes to Mycobacterium and also Vibrio. So in this way, we can use the diagnostic kit or diagnostic assay to help us in identifying what is the species infecting. So with the application of the small RNA in PCR assays, so we generate some diagnostic kits In this picture, I showed here for lepto, for S. typhi, and also for TB. Now, there are some publications on our work, non-protein coding RNA in Vibrio, in Salmonella, and also Mycobacterium tuberculosis. So in conclusion, new small RNA from model organisms and pathogens has been identified, and it opened opportunities to identify the role of small RNA in regulation, so including pathogenicity and also as diagnostic biomarkers. And we successfully produced several PCR diagnostic kits for pathogenic organisms. So this is our group, research group in AMDI under the infectomic cluster, and you can go and see our website, RNA Bio. Here I put the address. Okay, thank you for your attention.
Thank you, Doctor. Now audience can ask their question in the chat box or can unmute yourself and ask questions.
Doctor
Yeah.
Siti. Yeah,
Yes.
I actually have one question. So, Dr. Siti, regarding the functional characterization of those non-coding RNA, assay were used, right?
Yes.
Yes. Can you elaborate on like usually what type of assay is used? Like what type of assay is based on?
You can firstly you can use electrophoretic shift assay to see whether there's any binding with between small RNA or with whatever kind of proteins, such as other proteins that can bind to sRNA. So when we do the binding and we run through the gel shift assay with the binding of protein, it will move very slow compared to the one without binding. So this is one of the assay that you can use to identify what is the target. Usually we will predict first the function of the small RNA by looking at whether it's a antisense or whether it's a transacting small RNA. And we can also predict by looking at, comparing with the other pathogens that people have done before, if it's more or less the same like the small RNA that has been identified in model organisms. So we might be able to predict, and then after that we can run the appropriate test to see whether our prediction is correct or not.
I see, Doctor. I see. So it's mainly based on its interaction with the different type of proteins.
Yeah.
And also from previous research.
Yes, yes.
I see. Okay. Thank you, Dr. Siti.
You're welcome.
Doctor, there's the question. Is it non-coding RNA can be used to treat disease?
Now I am not sure whether we can use non-coding RNA to treat disease, but when we found small non-coding RNA antisense to a coding sequence. Let's say, if this, let's say the coding sequence codes for something that is involved in disease. So if we can do something with the small RNA that is antisense to this gene that is coding for diseases, we might be able to use it as a therapeutic line to treat disease. But I'm not sure about this, but I think it should be able to do the function, because when usually the small RNA antisense to a coding sequence. So meaning when it is antisense, when the gene is translate to protein, meaning that the small RNA is opposite. The expression of the small RNA is opposite, is down-regulated. But if we increase the, let's say, we increase the expression of the small RNA, it will, I think it will decrease the expression of the gene that is antisense of it. Yeah, it should be able to do, but you have to try and see to do this in an experiment and see whether it works that way. Yeah.
Okay, the next question is there any non-coding RNA yet to be discovered?
Yes, there are a lot of small non-coding RNA, and every day more and more research has found that people know more and more about this non-coding RNA, which is last time was not known or known as junk. People didn't know that actually this non-coding RNA has functions in the gene. That's why, and the research in non-coding RNA is still a lot to go. Yeah, there are many non-coding RNA that are not characterized. Function is unknown, and yeah, you can always try to embark in this field of non-coding. Yeah.
So the next question from the audience is, may I know how the PCR diagnostic kit works?
So okay, we have identified a small non-coding RNA. Let's say I go back to my slides just now. Small non-coding RNA. Let's say we found it in Salmonella typhi. But then when we see it, this small RNA, this particular small RNA, is not it doesn't exist in other in other types of Salmonella. Okay, so by using this small RNA gene, we can design the primers to identify these small RNA genes that is specific to S. typhi, let's say, and we can put the primers that can detect this small RNA in a PCR. So when you have S. typhi, it will generate a band for S. typhi and not for other Salmonella. So in this way, we can differentiate between the Salmonella species. Yeah, if I explain it correctly. Yep.
So there's one last question from them.
Yeah.
Is there any research that relate to non-coding RNA that you are currently working on?
Yes. Now, yeah, my PhD work is in identification of small RNA in Leptospira interrogans. Well, actually, what we did was transcriptome of the um, leptointerrogans. Transcriptome is a study on all the transcript in lepto. So we found that, you know, so many new small RNA, and we also identify the because it is a transcriptome study, so we also map the transcription start sites. So there are many potential sRNA candidates that is still need to be characterized and to know the function in this leptospira, how this small RNA regulates the leptospira either in pathogenic leptospira or in non-pathogenic leptospira. So what are their functions and how they regulate the pathogenicity of the leptospira? Yeah.
Thank you, Doctor, for answering the questions. Thank you for giving
You're
this
welcome.
opportunity to me to serve as moderator. Thank you, Doctor, for joining this session.
Yes, no problem.
Thank you very much, Dr. Siti Aminah Ahmed, for your wonderful talk. And thank you very much, Ms. Pravina, for moderating the section. Now, I would like to invite Ms. Ziching for vote of thanks.
Thank you, Ms. Montaj. Hello, everyone. I'm Dazi Ching. I'm proud to tell you that I'm a Year 2 student of BSc Honors in the Biotechnology degree programs of AIMS University. The organizing committee has given me this wonderful opportunity. First of all, on behalf of the ABSA and Webinar Organizing Committee, I want to thank Dr. Siti Aminah Ahmed for an enlightening and informative talk on insight to the non-coding RNA world. Dr. Siti Aminah Ahmed, we are grateful for the time and effort you took to share your thoughts and experience with us. We really appreciate it. Thank you to Senior Professor M. Ravichandran, Dean of Faculty of Applied Sciences, for showing your support to us by attending today's webinar. I want to thank Mr. Hugo, President of ABSA, and Ms. Vivian, Vice President of ABSA, for their help. I would also like to thank all the participants from various countries for joining us for this webinar. I believe that this webinar definitely helped you to get some insights and learn something new about inside the non-coding RNA world. I would like to take this opportunity to record my thanks to all members of the webinar organizing committee for their hard efforts. Working together as a team for this webinar was really engaging and enjoyable experience. Last but certainly not least, my special thanks to our ABSA advisor, Mr. Jivendran, for his continuous support as we learn lots of things through this webinar organizing exercise. Ladies and gentlemen, once again, thank you. Ms. Montaj, over to you.
Thank you, Ziching. It was a great section today. Before ending this section, we would like to hear feedback from all participants by scanning the code on the screen right now. We promise the evaluation survey would not take more than 1 minute. Your feedback will be greatly appreciated. You can either scan the QR code or go to the link shown below. The link is posted in the chat box as well. After the completion of the survey, you will receive an instant participation certificate in a short while. And I will keep this slide running for 2 more minutes for you to scan the QR code. We're looking forward to have you all joining us again in the next section. Thank you again, everyone, and we wish you good health and safety.
Okay, bye-bye.
Miss, do you want to stay back first? Maybe we can snap a group photo after this.
Huh? Group photo?
Yeah, I mean virtually. Yeah.
Okay.
Virtual group photo.
Okay, okay, sure, no problem.
Yeah, okay, Miss.
I,
Once
I,
again, thank you for
Actually,
coming.
this is my first time doing this. I am the one who usually listen to a webinar or something, but I've never done a talk. mm, giving a talk through webinar before. So this is my first experience.
That's good.
I see.
Yeah, good job.
Siti. It was very simple and nice. Very, very good.
Yeah, really informative. Thank you.
Yeah, welcome. Okay.
Dr. Siti, you're from ADMI, right?
Amdi.
AMD. It's
Oh,
Advanced Medical and Dental Institute.
I see.
USM. Yeah.
Yeah. Dr. Siti, do you happen to know Dr. Komita?
Yeah. Yes. She's also from infectomy clusters. Yeah, I know her.
I see. Since she actually came to our campus to give us a talk on CRISPR-Cas9, I think 2 years ago.
Oh, okay. Yes.
Yeah.
She's doing something using CRISPR-Cas9 on HIV, I think.
Yes, I heard. I remember. And I believe like some of our year 3 students is currently applying for AMD as well to do their internship industrial training.
Oh, yeah, it's good. It's good.
Yes.
Yeah, actually, we are kind of how to say, AIMS is one of our feeders for postgraduate students, actually, because AMD, as a research institute, AMD doesn't have undergraduates. So we don't have undergraduate students. So that's why sometimes we rely on training students that comes from AIMS doing their training, and then after that further their studies with us. So some comes from Universiti Sabah and do training with us, and then after that decided to join us.
I see, Dr. Siti.
Yeah. I think quite a number of AIMS students come, and there's 2 students now under Dr. Sitatan. I think they are formerly AIMS student.
Yes.
Devendran and Naveen, I think. Yeah.
Alumni.
Yeah.
I hope everyone had the chance to scan the QR code. Thank you for joining us in our webinar, and I'm going to end the meeting. See you soon in our next webinar. Stay safe and have a wonderful day. Until bye for now.
Bye.
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 So I can leave now, right?
Yeah. Thank you, Siti.